ELISA stands for Enzyme-Linked Immunosorbent Assay. It's a technique based on the detection of proteins using antibodies, and it has been successful in detecting a variety of diseases such as influenza, HIV, and SARS. So how does it work, you might ask? In your own body, your immune system makes antibodies that attach to antigens, which are markers on foreign invaders such as viruses or bacteria that don't belong in the body. As a result, antibodies identify and stick to these invaders in order to mark them for destruction by other parts of the immune system. So you can say that they're pretty good at finding antigens, and thus they're used in ELISA to find and mark antigens for our purposes. There are 4 components that constitute the mechanism of ELISA. Antigen, which is the target of the test, usually identifiable parts of the virus, bacteria, etc., and that's what we want to test for. Primary antibody is the antibody that binds directly to the antigen, and these can be produced by injecting the antigen of interest into an animal and then harvesting the antibody from the serum. And secondary antibody, which basically attaches to the primary antibody and has an enzyme that causes a color change with the addition of a substrate. And last but not least, the substrate, in presence of enzyme in the secondary antibody, as I mentioned earlier, and this causes the color change and tells us that ELISA detects the antigen in our sample.
An ELISA assay is usually conducted using a plastic microstrip tube that contains 12 wells. Each strip has designated wells for a positive control, a negative control, and 2 patient samples. Each of these 4 samples will be replicated 3 times to ensure that the results are reproducible. We will be using a blank sheet of paper to model what occurs in a well within a microstrip plate. The first step of this assay involves binding the antigen to the well. The patient sample is added to the well. Note that the patient sample contains many different proteins that may or may not contain the antigens we are trying to detect. Also note that the proteins we are trying to detect bind non-specifically to the plastic well because of hydrophobic interactions. Just for reference, non-specific binding means that an antibody is binding to unintended proteins, receptors, or transporters. Once the patient sample is added to the well, we must add a wash buffer to remove anything that isn't bound and block the inner surface of the well. This will prevent anything from binding non-specifically in future steps. And as a precaution, this wash step is repeated to ensure that we've got everything.
After we bind the antigen to the well, we'll need to bind the primary antibody to the antigen, which is the second step of this assay. To do this, we'll add the primary antibody to the well. The antibodies only bind to a specific antigen out of the many antigens that may be bound to the well. And this is why antigen-antibody interactions are specific and strong. After adding the primary antibodies to the wells, we'll need to incubate the samples for 5 minutes and then tap the fluid. We'll then add wash buffer to the well to rinse out anything that isn't bound. So if the primary antibody did not bind to any antigen, it will be washed away after we add the wash buffer. And we'll repeat the wash step one more time to make sure that all the unbound primary antibodies are washed away.
The third step of this assay involves binding the secondary antibody. The enzyme-linked secondary antibodies are added to the well. Note that these antibodies bind tightly to any of the primary antibodies that are present. This is because the secondary antibodies are covalently linked to an enzyme, most commonly referred to as horseradish peroxidase, which will produce a color change in response to catalytic activity. We'll incubate the sample for 5 minutes and then tap out the fluid. We will then add wash buffer to the well to remove any secondary antibodies that did not bind. As a precaution, this wash step is repeated 2 more times to ensure that everything is gone.
After binding the secondary antibodies in step 3, in step 4, which is the last step, we'll add an enzyme substrate to detect the antigen. After adding the enzyme substrate, we'll wait for 5 minutes and then evaluate the results. If there is no antigen, then no primary or secondary antibodies will bind, so the well will remain clear. If the antigen is present, then the primary and secondary antibodies will bind, and the well will turn blue. And that is how we are able to detect different diseases and conditions using ELISA.
ELISA can also be used to detect antibodies in a patient sample. An example would be COVID testing, in that we're not looking for antigen, but rather we're looking for the presence of certain primary antibodies. When someone is already infected with coronavirus, their immune system makes antibodies to viral proteins, namely S protein. Thus, we would add lab-purified coronavirus S proteins the appropriate secondary antibodies, and the substrate for our secondary antibody. In this case, if the primary antibody attaches to this protein, and thus coronavirus is present, then we can observe a color change which shows us that we got a positive result for coronavirus.
Thank you for watching.